Hello, dear judges. My name is William, and the topic of my research is a novel holdfast marking behavior for seahorses. We all know that dogs urinate to ward off other individuals, but not only dogs do so. In fact, a huge variety of terrestrial animals mark this sight or trait. However, in the marine world, examples of marking behavior have not been reported and observed yet. Many animals, no matter terrestrial or marine, show sight fidelity. In other words, they tend to stay in a fixed area whether it is its breeding season. This made me wonder, how do marine animals show sight fidelity? After paper survey, I found out that Hippocampus whitei, which are a kind of seahorses, are among the most sedentary of vertebrates. Since seahorses are poor swimmers, they are most likely to be found resting with their prehensile tails wound around a stationary object called holdfast. After doing more research, I found out that the longest period in Hippocampus whitei recorded on the same holdfast was 17 months for a male and 10 months for a female. Therefore, I chose seahorses, which are the representing species of sight fidelity, to start my research. When reading papers related to seahorses, I also found out that both adult and juvenile Hippocampus whitei had a strong preference for larger corals, especially avoid perching on small holdfasts. It occurred to me that there is major coral reef destruction due to the ghost nets tourists and anglers left. Hence, the aims of my study also include determining how the coral destruction will affect the seahorses population. When feeding the seahorses in my lab, I accidentally saw my seahorses secrete a kind of secretion, and this is a short film. As time goes by, the unknown secretion surprisingly drifts and attaches to the seahorse's holdfasts every time, and this phenomenon occurs once over 2 to 3 days. This is my seahorse defecating. These two kinds of secretions aroused my interest, so I started my research. My research can be divided into 3 segments: secretion investigation, behavioral investigation, and anatomical investigation. My first experiment is to do some microscopic examination in order to know whether this kind of secretion is a kind of feces. In this photo, we can tell that there is obviously structures of seahorses' food, crustaceans, in the frame. However, when it comes to the unknown secretion, there isn't any structure of their food in it, and no matter what kind of food I fed the seahorses, the unknown secretion remained mucus. On the second experiment, I analyzed the drifting pattern of feces and the unknown secretion. I cautiously put the 2 secretions in a beaker filled with seawater, then I used image J to analyze my results. After analyzing, it is shown that the time the unknown secretion drifted is up to 12 times longer than feces. Next, I recorded the secreting cycle of feces and the unknown secretion to know whether the process of the secreting is related to defecation. Here are the results. Decreasing the feeding frequency results in a decrease in the frequency of defecation, but the frequency of the secreting is unaffected. Therefore, it is now apparent that the unknown secretions are not feces. It occurred to me that the real secretion I found appears on the seahorses' holdfasts every time. So, maybe seahorses show sight fidelity by secreting this kind of substance. Hence, my next experiment is to observe that whether seahorses sense the unknown secretion and feces. This is my method. I prepared 2 water pipes as holdfasts, and I attached a piece of sponge on each of them. I put a piece of the secretion in one of the sponges, which let the seahorse can only sense it by olfaction. Then I let the seahorse choose a holdfast to perch on. I used chi-square test of independence to analyze my results and calculate the chi-square value and p-value. After analyzing, it is shown that the results are statistically significant. However, when I repeated the method on feces, the results are non-significant. I finally found the answer to the mystery. Seahorses sense the secretion by olfaction to show sight fidelity. Subsequently, I wanted to find out whether the secretion has the same trait recognized in other species markers, that is, its function will decline as time passes. After experimenting, I found out that the secretion does gradually loses its function, and on the 7th day, the seahorse shows no preference to hold fast with its secretion on it. So, I now named this secretion marker. I continue to investigate the key component of the marker by extracting the key component and using LC mass to identify the component which leads to my result. After extracting, I found that the key component is water-soluble, and it may have an M/Z of 185.115. We knew that dogs and cats urinate to ward off other individuals. This made me curious. Would the seahorses' markers have similar uses? Thus, I conducted an experiment to find out whether the markers have special meanings to other individuals. Yet, the results show that no matter the marker is from other individuals of the same sex or of the opposite sex, the result remains non-significant. This indicates that every seahorse's marker is only for marking its own site. I went for more papers, and surprisingly, the papers indicated that Hippocampus whitei may not actively defend their species' territories, which matches with the results of our experiments. So, the conclusions of this segment are: first, seahorses sense the marker by olfaction, and the marking effect of the marker can last up to 7 days. Second, the key component of the marker is water-soluble. Third, every seahorse's marker only marks their own site. It does not ward off other individuals or attract mates. Next, in order to find out which organ secretes the marker, an anatomical investigation was conducted. After dissecting 2 seahorses, a male one and a female one, I found out that the excretory organs of the markers are their gonads. However, when I did section cutting, there are no gamete cells found inside the gonads. This implies that the holdfast marking behavior is not relevant to whether the seahorse is sexually matured. To wrap up this segment, I found that it's the seahorse's gonad which secretes the marker, but the holdfast marking behavior is not related to the sexual maturity of the seahorse. In the marine world, examples of marking behavior have not been reported and observed yet. The holdfast marking behavior for seahorses I found is the first case. Maybe many marine animals which have site fidelity actually mark their own territory, but still not reported. I sure hope that my discovery and methods can provide a precedent for all scientists and help them discover more interesting behaviors under the sea. People started to focus on seahorses cultivation, but there is bad news too. Seahorses may go extinct if we still destroy the coral environment wantonly. Because of using inappropriate ways to fish, many tourists or anglers often leave ghost nets, which are fishing nets abandoned under the sea. This not only disables the corals to survive, but also destroys the habitat complexity and flattens coral reefs, kelp holdfasts, sea fans, etc., which deprives the seahorses of large holdfasts. Since the seahorses mark their own holdfasts by ejecting a kind of marker, the less complicated the holdfast structures are, the more difficult it is for the markers to attach on the holdfasts, which make it impossible for the seahorses to show site fidelity. After that, it will be harder and harder for seahorses to camouflage well and rear offspring, and eventually lead to seahorses' extinction. Fortunately, there are some nature reservations in Australia, which ensures the prosperity of seahorses dwelling in the area. In the future, I hope that there could be more and more seahorse reservation sites all over the world to return a better world to them. My study clearly shows that breeding seahorses simply could not save the species. If there are not enough large holdfasts on which to perch, seahorses will eventually become extinct. My study also gives a new impetus to seahorse conservation by showing the importance of protecting the entire coral reef habitat and rebuilding complex habitats. Coral reef habitats must be preserved or artificial holdfasts could be provided for seahorses. This would allow easier marking of holdfasts and increase the chances of seahorses' survival. This is the end of my presentation. My name is William, and the topic of my research is a novel holdfast marking behavior for seahorses. Thank you very much for your attention.